Hello everyone, welcome to the second live session of the first International Electronic Conference on Entomology, IECE 2021. My name is Mina, I'm from MDPI and I'll be your host for today. This e-conference is hosted online from July 1st to July 15th, 2021, and is organized by MDPI and the journal Insects. During this time, the conference presentations will be available for discussion on the conference's website. And the journal Insects will publish a special issue of the conference proceedings papers. I would like to thank the conference chair, Professor Nickolas Kavallieratos, the conference committee, and the session chairs for making this e-conference possible, along with the speakers and, of course, our attendees. I would like to remind everyone that this webinar is being recorded and the recording will be made available as soon as possible on the webinar's webpage. Today's session is also being live streamed on MDPI's YouTube channel, so hello to those of you watching us live. Today's live session will focus on pest management. There will be 5 presentations today of 15 minutes each. The presentations are followed by a question time moderated by the chairs. And if you have any questions for our speakers, please feel free to post them in the Q&A box at the bottom of your screen. Please also note the person to whom you wish to ask your question. I will now briefly introduce our session chairs. We have Dr. Rob Morrison, who is currently a research entomologist at the USDA-ARS Center for Grain and Animal Health Research in Manhattan, Kansas, where his current research program involves elucidating the chemical ecology and behavior and improving the IPM of stored product insects. We have Professor Dr. Brighton Mvumi, who is a professor of post-harvest science and technology at the University of Zimbabwe and has facilitated numerous post-harvest training programs for technocrats and scientists in SSA, and is experienced in the evaluation and optimization of pesticidal plants in agricultural pest management. And last but not least, we have Professor Dr. Jerry Zhu, who is a research chemical ecologist at USDA-ARS and is also a professor of entomology at the University of Nebraska. His research focuses on semiochemical-based pest management, discovering novel natural product compounds for practical applications. Thank you to all 3 of you for jointly chairing this session. I now hand it over to you. The floor is yours. Enjoy, everyone.
Thank you very much. And it is a really pleasure and to have the opportunity to organize and gathering all the really excellent researches and shared with all the advancement and development in pest management. And during this difficult time, and glad that we have a platform and to make it happen. I would like to thank my colleagues Brighton and Rob and who helped to put this session together. We received over 90 submissions and through a lot of filtering, and we selected the 5 best presentations and to share all the audience and attendance today. So I hope you will enjoy their work and enjoy their talk. Rob and Brighton probably will moderate all the speakers, and so we will give you about 12 minutes, maximum 13 minutes, and for your presentation, and then please leave about 1 to 2 minutes for discussion for quick Q&A session. At the 13 minutes level, and we will give some warnings like raise the hands, and so you have to wrap up your talk immediately or as soon as possible. All right, and then I will give the platform to Rob, and Rob, please.
Thanks, Jerry. Appreciate the introduction. So it's my pleasure to introduce our speakers today. First up, we'll have Dr. Deanna Scheff, who's with us from the USDA-ARS She's a research entomologist in Manhattan, Kansas, and currently works in the Stored Product Insect and Engineering Research Unit on integrated pest management of stored product insects. She'll be talking today to us about evaluating the practicality of spinosad for use in packaging materials. Deanna, you want to go ahead and share your screen with us and take it away?
Wonderful. Thank you for that introduction. And I will get my screen shared and pull up that presentation. All right. Well, thank you all. Good morning, good afternoon, good evening to everybody that is attending this presentation today. This is such a great opportunity to present a little bit of the research that I've been working on over this past year, looking at evaluating the practicality of spinosad for use in packaging materials. So to kick us off, to get us started, I'm going to talk just briefly here about the global post-harvest system. And this may be familiar for some of you and it may be kind of new for others, but this is kind of a generalized visualization of kind of what post-harvest system means. So when we think of post-harvest, we think of basically once that wheat, corn, soybeans are being gathered from that field, they're being harvested from that field. That's really where this supply chain starts. So after grain is harvested, it's generally transported and stored in either some type of metal silos or in storage bags, depending on different areas of the United States and all over the world. After that storage period, that grain product is going to go through a processing step. So here, for example, we have a flour mill where we're milling down wheat. So that wheat is going to get milled. After that milling product, it's generally, again, transported someplace else. It's packaged and ready to be sent out on trucks, on barges, on railway, eventually all the way to that market, right? So eventually to the marketplace where you and I buy it or we sell it to other consumers. So what's really interesting about this global post-harvest system, no matter which area that we see within this chain, food loss occurs at every single step in this supply chain. So food loss can be due to spillage, so that grain truck hitting a bump and grain falling out of that truck. It can be due to spoilage, more microorganisms, if we're not properly drying down that grain while it's being stored. It can also be due to insects as well, right? So in our milling and our warehouse system, there's always an ever presence of insects and they can contaminate that food supply as well. So overall, it is estimated that nearly 1/3 of food that is produced is lost throughout the world. And specifically, if you look at developing countries, it is estimated that over 40% is actually lost post-harvest. So after that grain is coming out of that field before it hits our food plate, that we're losing food. And what we really want to focus on are what are ways that we can help reduce some of this food loss system. And one area that I'm really interested in and have been conducting research in for quite a few years now is this idea of the packaging and shipping aspect. And specifically, if we look within this global post-harvest system at packaging and shipping, we can see that we do lose food for insect infestation of those packaged food products. So insects are chewing through that packaging material or invading through broken packaging. We also have food loss due to poor pest management techniques, again, with stored product insects, but then also looking at birds or rodents as well. If we're not taking care of those other animals, we're going to have some food loss in there. Again, we also have those transportation failures and then failure to maintain proper storage conditions. So creating an environment where insects thrive or even fungi thrive as well. So we want to kind of limit those things to be able to harvest our food, store it, package it, and sell it, and make sure that we can maintain that overall food safety. So I'm going to focus a little bit in on this relationship of actually insects and packaging. When you think about packaging, the primary goal, regardless of any contents that are being stored in there, but that primary goal of food packaging is to delay that product deterioration, extend shelf life, and maintain the overall food safety and integrity of that food product being stored. So one tool that we've been investigating at the USDA for a number of years right now is this idea of insecticide incorporated packaging. So currently we actually have one type that's available in the United States. Specifically, this is a methoprene-based packaging material. And methoprene is a great tool to be able to incorporate into packaging because it's an insect growth regulator. So it's very specific to what it affects with that insect, with that juvenile hormone, which makes it safe to use for our food packaging because it's overall has very, very low mammalian toxicity, excuse me. But it's also very effective on our juvenile stages of our insect species. So we've done numerous studies throughout the years with different insect species that we can see significant reduction in adult emergence on larvae exposed to this packaging material for short-term or long-term periods. We've looked at all types of store product insects, including red flower beetle, warehouse beetle, larger cabinet beetle, and then even this invasive species, capra beetle or Trochoderma granaria. As we can see over here on the right, so we were looking at, in this study here, we looked at craft paper, woven paper, and then two types of polymer paper where we incorporated this methoprene inside that packaging material. And then we do see significant reduction in adult emergence on our different types of packaging material. And generally, those polymer packaging work a little bit better than our craft paper or more woven type packaging material. So this is great. We've got some knowledge base on this methoprene packaging, but what's really interesting is what else can we look at possibly incorporating into this packaging material? And the idea of spinosad came up. So spinosad is an insecticide that is derived from the fermentation products of a bacterium. So it is what we would call an organic product. So it opens up a whole nother marketplace for us to use this insecticide incorporated packaging. Spinosad is also highly active, so it works either by contact or ingestion to a variety of pest orders, including Lepidoptera, Diptera, Hymenoptera, and then Coleoptera. There's been numerous amounts of research conducted with even store product insects, and it's found to be very effective when spinosad is applied as a grain protectant to grain that's being stored for long term in these kind of silo areas. There's been a few studies looking at the application of spinosad onto a concrete surface or a metal surface, and what we've found is that generally the oral toxicity is about 5 to 10 times greater than having this contact toxicity. So it's interesting that this product works really well when it's injected, not as great as a contact insecticide, but it also still has that propensity to work as a contact insecticide. So our objective was to investigate this packaging incorporated with spinosad alone, or also in combination with the traditional methoprene across a variety of product insects. And for this presentation, and I'm just going to focus specifically on Trochoderma variable or the warehouse beetle. So with this, we worked with a collaborator in the United States called Provision Guard, and they were able to create cardstock and polymer packaging for us. So cardstock, as you can see up here on the top, and then a polymer-based packaging here on the bottom. So we created arenas on the bottom of a petri dish with material that was spinosad only, methoprene only, or a combination of spinosad and methoprene as our test treatments. And then, of course, we use filter paper for our control material. So we looked at both Trochoderma variable, adult and larvae. So adults we placed on our packaging material here, and then we looked at adult mortality. And then for larvae, we placed them also on that treated packaging material and then held them for adult emergence for roughly 4 weeks to allow for the larvae to make it to the adult stage. What we can see is actually kind of very interesting between the two different life stages of this insect. So our first test with this cardstock, we're looking here at adult mortality. As you can see, over over a week span of 7 days, we saw no significant differences in adult mortality, whether it was on the treated packaging or on our control packaging. We do see a slight increase at 7 days. Again, that's not significantly different from our control packaging, so it's most likely just due to natural mortality of these beetles since they are short-lived. However, when we look at what happens with our larvae, right here we can see our combination packaging had 0% adult emergence, whether they were normal adults or a deformed adults, as shown up here. Our methoprene only had roughly about 20% adult mortality, but our spinosad packaging only actually was not significantly different than our control. So what we're seeing is that there's something in there with that combination packaging of methoprene plus spinosad, but spinosad only by itself was not very effective. The methoprene is behaving as what we normally expected, but this combination packaging, what was interesting is that we had roughly about 48% of our larvae still alive after 4 weeks. So they're not progressing to that next life stage, which is a positive attribute. But even those that do potentially make it to that pupal stage, we're having these pupae, roughly about 38, just under 40% of our pupae are deformed pupae, and they actually die. So these are a couple of examples of what our deformed pupae look like. They have bits and pieces of adults starting to develop, but then they also still have that larval body inside that larval casing. And again, like I said, they all die. They don't make it to that adult stage. So we're very, very effective with this combination packaging. So then when we're looking at our polymer packaging, just a slightly different kind of matrix for inclusion for this spinosad. Similar to our cardstock, we actually don't see any differences in mortality after 7 days. And that's a little discouraging, but again, when we actually look at that larval aspect, is that we are very effective at that larvae, at controlling that larvae from making it to the adult stage. And here, we actually, with the spinosad, do see about a 38% adult emergence, but that is significantly lower than the control. So that's roughly about 50-ish% reduction in adult emergence, just using that spinosad only. So we are seeing an effect of that spinosad when we incorporate it into this polymer packaging material. We also see very little progression of those larvae to that next life stage with our combination and methoprene packaging. And even in our spinosad packaging, we still have quite a few of those larvae still in that larval stage. So we are disrupting that life stage progression of these insects to moving on to that adult stage. So that is very effective for what's going on with this polymer packaging. So kind of what we can kind of conclude a little bit about some of these preliminary studies is that we do see some differences in susceptibility between the larvae and the adult species. Our larvae are more susceptible, so it could be due to a little bit longer exposure. It can have other biological factors between adults and the younger instars, or it could be due to having some of that food being on that material and the larvae ingesting and having a little bit of that effect of that ingestion. We have differences between our cardstock and polymer packaging, and then that could be due to our application method of how that insecticide is actually incorporated into the packaging. But it's interesting to see that we are getting a little bit of that synergistic effect of that combination treatment between the combination of methoprene plus spinosad. So this is a great tool, both the combo and methoprene, that can be used as a preventive measure to help reduce the potential of infestation of packaged products. But however, we still need to do some more additional research on the spinosad-only packaging. This is just one snippet of one insect that we looked at, but briefly, we also looked at another species, Rhizopertha dominica. With this insect, when we had adults looking at that same mortality, we had 60% on cardstock and 92% on polymer. So it's working great on the Rhizopertha, not as good on the larvae or the warehouse beetle, or excuse me, adults on the warehouse beetle. But with that, I just want to say thank you to my collaborators, Dr. Jim Campbell and Frank Arthur at the USDA, my lab tech and student, Brian and Molly, Provision Guard for providing this packaging material. And thank you all for attending today, and happy to answer any questions. And then also my contact information there as well if you want to follow up with an email. It's well. So thank you so much.
Thanks, Deanna. I have just one question in the Q&A box. Claire asks whether you have information on the toxicity for pollinators for spinosad, or whether data only exists for pests.
Yes, I know it does exist. I don't have that currently right here in front of me for the spinosad. At least for right now, when spinosad is being used, it's used mainly in the grain protectant area, so it's not as open to pollinators. But I do know it's something that's in the background when it's developing this product. But I don't actually have that information in front of me, but I do know it does exist on that as well.
Okay, and then just one more question. Are there any further study on residuals on food that are planned to be done?
Yeah, so actually we're just developing a method with a colleague of mine that's a chemist. So we're doing some residual studies with food absorption of methylbrine. So finishing up developing that methodology, and then we're hoping to do it again here with spinosad, and actually a couple of our other insecticides that we do use to kind of see what that food potentially is grabbing from that surface, and then anything that the insects could eat and ingest that could infect them as well too.
Okay, great. Thanks, Dan. I appreciate it.
Wonderful. Thank you, everybody.
All right. Up next, we have Rakesh Kumar, who's a PhD research scholar at the Department of Biosciences and Bioengineering of the Indian Institute of Technology in Roorkee. He'll be talking today about the identification of small molecular inhibitors for Helicoverpa armigera. So Rakesh, you want to go ahead and share your screen and go ahead and take it away?
Yeah, thank you, Rob, for a nice introduction. So good morning, good afternoon, good evening to everyone. So myself, Rakesh Kumar. So today I will be talking about the identification of small molecule potential inhibitors for the notorious pest of that we call Helicoverpa armigera, and by targeting the enzyme that is GHMT, known like the juvenile hormone acid-methyl-transferase, through the in silico approach using molecular docking and molecular dynamic simulation. So I will start with the brief introduction of juvenile hormone acid-methyl-transferase. I am sure you all must be aware of this enzyme. This enzyme is a very potent enzyme of the juvenile hormone biosynthesis. It is actually this juvenile hormone biosynthesis starts from the mevalonate pathway, and then it branching out using the GH specific branch. In that, the juvenile hormone acid-methyl-transferase, it acts on farnesoic acid, and it methylates the farnesoic acid to methyl-transferase, and later on the methyl-transferase converted to juvenile hormone acid. That is the case of Diptera, Orthoptera, and Dipteroptera insects. But in case of Lepidoptera, the order of this farnesoic acid to juvenile hormone acid is like that is the order dependent. So these are the few reports that has already been published showing the importance of the juvenile hormone acid-methyl-transferase in the insect metamorphosis, the reproductive maturations. Here you can say some of the studies saying by the knockdown of juvenile hormone, acid-methyl-transferase enzyme in Lepidoptera, giving around 30 to 30% mortality. And similarly, in the Aedes, the yellow fever mosquito, again the RNAi-mediated silencing, it resulted in the larval growth and egg development. It is severely interfered. In one more study using the same enzyme being targeted using RNA interference in again the Tribolium castinium precocis metamorphosis has been observed. And in the targeted pest, that is Helicoverpa armigera, that is the recent study like in 2014 from the Indian scientists, where they have specifically targeted the juvenile hormone acid-methyl-transferase using RNA, and they showed here in this picture, they showed how the silencing has resulted in the deformed development of the larva. So keeping in view of these already published literature and the importance and the important role is being played by the gene hormone acid nucleotide transferase enzyme in the insect physiology. So I thought like let us target this enzyme for the development of some specific you know inhibitor by mining the data from the natural products, because the natural product obviously they will benefit by the they will be eco-friendly because they won't add up to the the soil or water pollutions as the normal insecticides do. So in this direction we have collected the insects and we have isolated the RNA, and using that RNA we extracted the juvenile hormone gene using the PCR gene specific primer, and we cloned the juvenile hormone gene into the bacterial cloning vector, and we sequenced it, and the sequence was confirmed and we got this sequence. So this is the sequence of juvenile hormone acid nucleotide transferase from Helicoverpa armigera. And using this sequence, since no crystal structure or the three-dimensional structure, either using X-ray or the NMR, is available, so we have to go for the comparative modeling using molecular modeling approaches. So we generated a three-dimensional homology model using this sequence, and we got you can see in the right hand side the homology model of juvenile hormone acid nucleotide transferase, and then we use this model for the validation whether this model was like perfectly okay to proceed with it. So Ramachandran plot is one of the tool. Though there are so many other tools are also available by which we can validate the model quality, but Ramachandran plot is one of the tool that I am showing here. According to this Ramachandran plot, we can see majority of the around more than 98% residues are in the favored regions. This is one of the like plot which gives the stereochemical quality of the proteins, whether the the main chain bonds and the side chain conformations in the model proteins are in good agreement with its stability or not. So here I am like drawing your attention toward uh, the the critical residues which has been already like identified in like some other insects, whether like the residues which are important for its activity of this juvenile hormone. Since juvenile hormone acid methyl transferase, since the methyl transferase, so it has two substrates. The one substrate is the same, that is the methyl donor. We call it S-adenosyl methionine. So the upper red residues which are polar in nature, so they are responsible for binding of the same moiety. And the blue amino acids, you can see these amino acids are mostly hydrophobic in nature. So they form the hydrophobic pocket, and that hydrophobic pocket, it forms the substrate binding pocket. In the down, you can see this is the hydrophobic pocket, and these are the hydrophobic residues which are surrounds this hydrophobic pocket. Because of the hydrophobic nature of its substrate, like the farnesolic acid is mostly hydrophobic in nature, juvenile hormone acid, except that the terminal COH group in both the substrate, everything is like hydrophobic in nature. So obviously this enzyme definitely like harbors the hydrophobic pocket. So this hydrophobic pocket was like it was predicted, or you can say it was confirmed using the homology modeling whether this hydrophobic pocket is there or not. So after confirming this pocket, so we search the compound library database for the mining of the compounds which are already available through some whether they showed any insecticidal or like the binding affinity for the juvenile hormone acid nucleotide transferase. So we search the measure the food database which has the collection of the compound from the mostly the fruit like crops and the fruit plants, and the Zinc 20 again it is a very big database. It contains all kind of molecule. So we use this database, but mostly I restricted myself for the natural compound discovery. So we selected the food database, and using the chemoinform like the open source K9 software, the molecule from this database was screened for the very well Lipinski rule of 5. The Lipinski rule of 5 says, like, it identifies the molecule, or you can say it filters the molecules as per the drug-like criteria. So Lipinski says, like, if a molecule with less than 500 delta of molecular mass and less than 5 number of hydrogen bond donors and less than 10 numbers of hydrogen bond acceptors and the water octanol partition coefficient of less than 5. So these kind of molecules will like they increases the chance for the like permeability or the absorption within a system. Though this criteria is utilized for the drug molecule which are going to be used in the animal research, but definitely this small molecule will also help in our insect system. So I proceed with the virtual screening. I have prepared the molecules, the ligand molecules and the protein molecule by adding the partial charges, and their geometry was optimized, and the active site was already specified. So I prepared a grid molecule around this active site, and I proceeded with the docking, and the docking result gave me like these 4 main compounds: this scolimoside, luteolin, hormonectin, and picrocrosin. These are the compounds I got it from the food database, and these are like compound number 5, 6, and 7. They are mostly from the cotton metabolite only. Beta amyrin, beta cytosterol, gossypol. Everybody, you know, that is a aldehyde molecule abundantly present in the cotton plant, and helioside B1 also. So, and these the last 2 compounds are junyl hormone acid and the farnesoic acid. These are the natural substrate. So you can see by the docking, the affinity like scoring, I got around 6.2 or 5.9 for the natural substrate. But for those compounds, I got like more than this. So this compound, the scolimoside, I got the 9. It means it has at least 3 times more affinity than the natural substrate. So this can be like considered as a good candidate for the further study. So here in this picture, you can see this is the active site. You can see all the ligands that are selected for the virtual screening or they come out of the virtual screening. They are like sitting in the substrate binding pocket. Here you can see the substrate molecules are sitting. Here the screen compounds are sitting in the same substrate binding pocket. So it means these molecules are not binding anywhere in the proteins. They are targeting sitting particularly in the active site. So here you can see the interactions of the natural substrate, that is the farnesoic acid and the junyl hormone acid. They mostly, because since only the terminal groups are polar in nature, they are involved in the hydrogen bonding. But rest of the chain, like tail, is hydrophobic, so they are the forming the hydrophobic and the planar interactions with the hydrophobic amino acid residues. Similarly, the identified 4 compounds also showed because they are sitting in the same hydrophobic pocket. So, they are forming because of their the polar residues, polar functional groups. They are making both polar interactions as well as non-polar. But the binding residues, are one and all same in case of substrate as well as in the inhibitor. So when I search those compounds, like what are those compounds and how they how we can get it for the further study. The scolimosides, like I got, that is isolated from the weed capsula, that is commonly known as shepherd pus, and it is a minor flavonol that is extracted from the leaf and extract of artichoke, and other also the picrocrosin, that is also from saffron, and it's a monoterpene glycoside, and the luteolin again it's the digiflavonol, and the hormonectin is also isoflavonol. Means most of the flavonol compounds, they showed the affinity for the junyl hormone acid methyltransferase for the competitive binding for this substrate. So the docking results were, most commonly they are validated using the molecular dynamic studies. Yes, sir. Yeah. So the molecular dynamics, the first criteria is the RMSD. RMSD like says how much the deviation upon binding the ligand to the receptor, how much it is stabilizing or destabilizing the protein. Here you can see the complex, the general hormone complex with this colomoside, it is the RMSD get reduced compared to the free enzyme. It means upon binding to the enzyme gets stabilized, and the stabilization is also confirmed with the radius of gyration. The radiation of gyration, it is also given about the overall compactness of the protein. Upon binding of the ligand to the protein gets opened up or the closed one. So the reduction in the radiation of gyration says the protein gets stabilized upon ligand binding. And here also we can confirm that throughout the molecular dynamic simulation, the number of hydrogen bond formation and the breakdown, the average value was like 2 to 3 hydrogen bond, and they are constantly stable throughout the simulation process. It means the interactions are real. They are not random interactions. And once the ligand bind to the proteins, it get stick to the active site, and it is not getting disturbed during the molecular simulation. So best, because this is a very preliminary study and based mostly on the in silico ones, so I can conclude that based upon this is initial screening and this colomoside, and along with the picoprosin, luteolin and luteolin, sorry, this one permectinectin showed that reasonably good affinity for the JHMT compared to its natural substrate. So we can think like in future we can plan for the buying of or the extraction of these compounds from their plants, and we can test the in vitro binding affinity either through some biophysical techniques, like we have already cloned the proteins, and we can study some in vitro binding, whether they are really interacting in the experimental conditions, or we can feed those compound to the insect in an insect bioassay to test the efficacy of these compounds, whether they are really having any effect on the insect system or not. So since it is based upon the in silico, so I cannot that is also in the initial stage, so I cannot present the further data. So here this is my lab, this is my supervisor, Professor Ashwini Kumar Sharma, and rest of my colleagues. The downside is the building at Indian Institute of Technology. So thank you all for your kind attention. So I'm happy to answer the question.
Thank you for that, Rakesh. I'm looking in the Q&A. I don't see any questions. Seeing none, we'll go ahead and move on to our next person. Thanks again, Rakesh. I appreciate it. Our next speaker today is Fabiana Sassu. She, from 2016 and 2019, Fabiana held a PhD scholarship at the University of BOKU in Vienna. During this period, she carried out most of her work at the FAO IAEA Insect Pest Laboratory of Zebersdorf, where she developed most of her PhD project. Her project focused on the invasive insect pest Drosophila suzukii, with the main task to develop irradiation and mass rearing techniques for application of sterile insect technique. And after she graduated in March 2020, she's been now on maternity leave. So with that, why don't you go ahead and share your screen, Fabiana, and take it away.
Hello, everybody. Today I'm Fabiana Sassu, and today I'm going to present my preliminary result about the mating competitiveness of the irradiated Drosophila suzukii under different atmospheric conditions. Suzuki is an insect pest who has invaded US and Europe, and recently also Africa. Is able to lay the eggs in fresh rapid fruits, and the insects are quite distinctive. The males can be recognized by the black spot on the wings, while the female has the characteristic ovipositor, which is extended and with who is able to lay the eggs in fresh fruits, which are usually stone fruits, berries, and also grapes. The damage is also increased by the very fast life cycle of this insect pest, which can have more than 15 generations per year, And the females have a really high fecundity because they can lay more than 350 eggs per lifetime. So one of the techniques that has been proposed as a biological control for this insect is the sterilized insect technique. This technique is based on the macerating of the insect pest, which is macerated in large numbers. Then the flies, usually as pupae, they are sterilized using ionized radiation, and they are released in the target area where there is infestation of this pest. And the goal is that, that the sterile males will compete with the wild males to meet with the wild female, and so there will be a decreasing of the natural population. Today, I'm going to show you what is my study regarding this last step, so the mating competitiveness of the sterile males. So, but first of all, we have to apply irradiation protocol and atmospheric treatments. For Drosophila Suzuki, has been already published that the dose to sterilize adults is to expose the pupae has a dose of gamma ray of 220 gray. But during this process, usually the ionized radiation can damage the organism cells because of the free radicals, which are present and generated during the radiation by the oxygen present in the atmosphere. For this reason, then there is a decrease of consequence decrease of the quality of the sterile males. And in some cases, this can lessen the ability of the male to compete, so the mating competitiveness. So one of the tactics or the strategies that has been applied is to use a reduced oxygen level. So the pupae, we had treated the pupae in normal air condition, which we will call normoxia, means that they are not actually treated with any atmosphere conditions, but they are just irradiated in normal air with the normal almost 21% of oxygen. And also we irradiate flies which are in a low oxygen condition by sealing them in packages. And this atmosphere create hypoxia level of oxygen, which is very close to 0% of oxygen. So the mating competitiveness tests that I performed were set up in a way that we observe our flies from 9 to 3 p.m. And we observe different cross, but what we will be interested today is the cross where we have both sterilized and not sterilized flies. So in a cage, which is usually of the size you can see here in the picture, we were putting 10 irradiated males, 10 not irradiated males, and 10 irradiated and not irradiated females. And this is where three times this cross was performed. And of course, we also had a control cages in which we have no competitiveness, but only fertile flies. And today, what I'm going to show you is the preliminary result regarding the cross that we are interested to, which is the competitiveness between the sterile males and the fertile males to gain the mating with the fertile females. All the setup has also been repeated 5 times. Of course, since we have to place the same flies in the same, different irradiated and not irradiated flies in the same cage, I had to develop a way for mark this fly, and we were using dust. Pupae were dusted and with different colors, and following the formula you can see here above, below. And this dust applied on the pupa case will be then retained in a membrane which is expanded used by the flies to gain exit from the pupa case, that is called pilenium. And this membrane here, then of course by touching the pupa case, then retain in itself also a part of this dust, which is then visible under the UV light microscope. The observed parameters were 3: time to mating, time from the beginning of, which is the time from the beginning of the sperm to the beginning of the copulation, then the duration of the copula, which is the time intercourse between the beginning and the end of the copula, and of course the percentage of the mating, which was done by the sterile or fertile flies. The first result, you can see that it was not a real difference between the two atmosphere treatments. Either hypoxia or normoxia did not have any difference with the control where there was no competitiveness. And the same was by the fertile males, which were in this cross, and the sterile males. Similar result we also have for the duration of the copula. So this is usually one of the parameter which is can be, let's say, having some consequences by the irradiation. In some insect pests, they reduce or along the time of the copulation. But in our case, there was no difference of the sterile males compared with the fertile mates when there was competitiveness or when wasn't. And finally, the percentage of the matings. Here I'm showing you on the left the hypoxia treated fly and the percentage of the matings of the fertile males with the fertile females, of the sterile males with the fertile females. There is also here not a significantly difference, and the same is happening for the normoxia. So basically, when there was competitiveness, neither the atmosphere or the irradiation had caused any lessen of mating competitiveness. In a similar graph, you can see the same data, but in comparison with the control where was no competitiveness. As you can see here, there is a significant difference, which of course happening because here was not, let's say, there were not irradiated or sterile males. However, in general, all these results are highlighting that in our colony, which was used for this experiment, the percentage of mating are still very low because we are below the 30%. Which is a very low percentage in case of using this fly for an SIT propose. In fact, our flies are less competitive what it will be in the, or what is in the literature for normal wild flies. So in conclusion, irradiation atmosphere treatment did not significantly affect the duration of the copula and neither the time of the matings. The competitiveness tests show no differences in the percentage of mating between the sterile and the fertile males in both hypoxia and normoxia crosses. And overall, the result of this study showed that the radiation does not have an impact on the male's competitiveness and that the atmosphere treatment did not significantly improve the male performance. Thank you for your attention.
Thanks for that, Fabiana. Looking at the questions, I don't see that we have any questions. Looking in the chat here, don't see any in the chat. So up next.
Hello, Rob.
Oh yeah, Brighton.
Yeah, I see a question there, which is talking about the best release ratio, please.
Oh yeah. Someone asked whether you can share your contact info from Ohana Rodriguez. So maybe you could follow up with them, Fabiana.
If somebody is asking for the release ratio, it's something needs to be still developed. We don't have yet what will be, but of course, in case of this will be the data of the colony mating competitiveness, then the release ratio will have to increase compared with the wild population. We don't yet have a pilot trial where we have even the field cages mating competitiveness. So we cannot yet establish this ratio. But of course, it's needed in a part of the plan.
And then someone asked whether you can share your contact info. I don't know if you have an email that you can share, or
Sure.
if you feel
I will
comfortable
put here
giving
in
that
chat.
out. And then we have another question also here while you're doing that, about whether you saw any difference between results when you use the same technology in another strain of Suzuki.
Yeah. Oh, I know that in other fruit flies, there has been same problematic, because when you keep colony under artificial condition for a long time, you either have to refresh the colony, or you have to anyway keep track of what's your competitiveness level, because it can lower along the time. And I don't know now what is, for example, in Ceratitis capitata or Anastrefa, what are their percentages of matings in the artificial colony, but it's probably has been for sure published, because, and I know that they do this test periodically, so there must be data on it.
Okay, great.
But maybe if they call, sorry, because now that I'm reading strain, maybe they meant my fly, so Drosophila Suzuki strains.
Yeah.
This is, so sorry, I probably answered something else. No, so if the Suzuki strain, there are differences in different labs in America, but also in Europe. I don't have, I haven't read or any published paper about the mating competitiveness or any strain of Drosophila Suzuki so far. So I don't know.
And then I think we're at time. So again, thanks, Fabiana. Very good presentation. I think it generated lots of discussion. So thank you.
Thank you.
Okay, so up next we have Dr. Sonia Duarte, who's a researcher at the Institute of Agronomy at the University of Lisbon. Our last speaker of the session today is Cedra Ajmi, who has a dual affiliation with the University of Tunis, Faculty of Science, and the Natural Agricultural Research Institute of Tunisia. She'll be talking today about application of agricultural fertilizers and essential oils as an innovative alternative for the control of Ceratitis capitata.
Hello, everyone. Thanks. My name is Insaf Sadrawi Ajmi. From Tunisia, a PhD student in biological science. I have the honor today to tell you about my works entitled Usage of Agriculture, DAP, Fertilizer, and Eucalyptus Essential Oils Potential Attractant Against the Mediterranean Fruit Flies Ceratitis capitata, elaborated by Insaf Sadrawi Ajmi, Najib bin Ali, Abir Sultani, Abdallah Fajrawi, and Judah Medyouni bin Jma'a. Common introduction. Ceratitis capitata in Tunisia, Ceratitis capitata was described for the first signs in 1855. Mayfly Ceratitis capitata is among the major destructive pests of fruit worldwide, particularly in the Mediterranean region. In Tunisia, mayfly is a key economic pest, and citrus and various other pests. This figure illustrated the damage of Ceratitis capitata. The picture is a picture of adult Ceratitis capitata. This is a larva and the shoot of fruits. On the other hand, chemical insecticide current control program is mainly based in application of insecticides. However, the use is subjected to much controversy, which results in the destruction of auxiliary fauna, the increase in residue levels in fruits, and the resurgence of secondary pests, and the development of resistant strains. So essential oils could be suggested as alternative due to their insecticidal property. On the other hand, diammonium phosphate is the most used phosphorus fertilizer worldwide. Tunisia is ranked among the largest phosphate rock production in the world. In addition, diammonium phosphate exhibited an attracting activity towards Ceratitis capitata. Objective: this work aims to assess the repellent attractant potential and the contact toxicity of Eucalyptus cinerea and Eucalyptus maydeni essential oils for the combinations for the control of Ceratitis capitata. This work aims to assess the insecticidal potential of formulation based on the combination of DAP and Eucalyptus essential oils against adults of Ceratitis capitata. Material and methods. Material animal. To initiate Ceratitis capitata, raring infested fruits were collected from various fields of Gafsa oasis. Raring was conducted under the following controlled laboratory condition. Larvae were maintained on artificial diet. For plant vegetal, Eucalyptus cinerea and Eucalyptus maydeni aerial parts were collected from Ain Draham. Extraction of essential oils was subjected by hydrodistillation. The analysis of chemical composition of essential oils were realized by GCMS. Formulation based on DAP, diammonium phosphate, and essential oils were prepared by adsorption. Bioassays. The first one, repellency bioassays. Repellency bioassays of Eucalyptus essential oils and encapsulated essential oils DAP against Ceratitis capitata were tested. The paper with 8 centimeters were cut into the halves. Firstly, Eucalyptus cinerea and Eucalyptus maydeni were prepared by dissolving 0.1 and 0.2 and 0.3 microliter of crude essential oils in 0.1 milliliter acetone. Each solution applied to half paper disc. The other half of the filter paper was tested with acetone alone as control. The tested and control half were air-treated and enough to evaporate the solvent. Treated and untreated were attached to the fusioning this and placed in petri dish. This 10 adults were realized at the center. Observation of the number of insects present in both the treated and untreated halves were recorded after 1, 3, 5, and 24 hours for each test control. And calculated the percent repellency according to Mvumi بنجمعها AR 2012. The second test was contact toxicity bioassay, contact toxicity of Eucalyptus essential oils, contact toxicity bioassay, contact toxicity of Eucalyptus essential oils and encapsulated essential oils and DAP were tested against Ceratitis capitata adults according to Salonki et al. 2005 with 3 repetition with 3 concentration. Results. Results showed that Eucalyptus cinerea and Eucalyptus maydeni were found to possess similar composition. The major common compounds were 1,8 cineol, alpha-pinene, alpha-terpineol, and alpha-confet. For chemical composition adsorbed, the results revealed that the major compound adsorbed for Eucalyptus cinerea and Eucalyptus maydeni were 1,8 cineol, alpha-pinene, alpha-terpineol, and confet. So adsorption selectivity was influenced by the abundance of each compound in the crude oil and the terpenic constituting polarity. For bioassays, test repellency. This figure showed that Eucalyptus cinerea and Eucalyptus maydeni were attracting towards Ceratitis capitata. This attractant increased with the augmentation of concentration. For both formulation, Diapi Eucalyptus cinerea and Diapi eumydini. Attractiveness were minus 66.66 per, and for Eucalyptus eumydini, percentage repellence equal minus 73.33. Alpha Diapi exhibited percentage également 20. For contact toxicity. Contact figure illustrated the percentage of mortality of mayfly adults exposed to crude essential oil, Eucalyptus eumydini, and eucalyptus essential oils and their formulation. Results show that the mortality increased with the increase in concentration and exposure time. For Eucalyptus crude, eumydini, and Eucalyptus cinerea. And the results revealed, on the other hand, that adults were sensitive to Eucalyptus their formulation exhibited a high percentage mortality against Keratitis capitata. Conclusion. And the signal was the major components for the both essential oils. The absorption of components were influenced by the major component in the determining components. The results from this work revealed that Eucalyptus eumydini essential oils crude and were formulated by absorption onto Diapi fertilizer was more attractive and more toxic than Eucalyptus cinerea. So results from this work revealed that the formulation based in the combined use of agricultural fertilizer, diammonium phosphate, and essential oils, Eucalyptus cinerea and Eucalyptus eumydini, could be involved as a compound for mayfly control strategy in oasis. This could be proposed aimed the current method for an EPH approach. Technology. This project is carried out under the MOBI scheme funded by the Ministry of Higher Education of Science Research through the PROMISE project and managed by the ANPR. Thank you.
Great work there. All right. Any questions for our speaker? I don't see anything else. So looks like that concludes our speaker portion of this. I want to thank again all our speakers. We've looked at a variety of management strategies today, including packaging, sterile insect technique. We've looked at how insects may be vector or interacting with microbes in the environment, and we've looked at natural products as well as some theoretical approaches.
Yeah, and Brighton, you want to give a short, brief summarization and on this session?
Yeah, yes, I can do that briefly. Thank you very much, Rob. Thank you very much, Jerry. We've had quite some wide range of topics covering the 5 continents. We also got some very interesting science, some of it which is quite cutting edge, ranging from stored product research, going up to, you know, field pests, molecular techniques, and also even the last one. And all these topics, they are addressing key pests that are competing with human beings for food, but also looking at the latest techniques or how those pests can be managed. I think the biggest challenge that I kind of draw out of these presentations is how can we take the science out of the lab to the field. In other words, how can we convert all these findings, these interesting investigations? How can we then take it out to the users so that the science is actually applied in real life? I think that's where we need to take the science to, so that we get it used. Otherwise, there's also a risk that the science remains in the labs or it remains in the journals. That is my major comment. It's very important to investigate and establish all the interesting facts and make sure that when it's then applied, there's a very solid basis for that. There's a very good understanding. So we wish all the presenters, the best as they build up their investigations, and hopefully we'll see.
It sounds like Brighton was cut off, and but anyhow, yeah, he brought a very important topic, and how we can use the knowledge and understanding from the basic understanding and then apply them into the practical problems which existing almost everywhere.
Wow.
So, go ahead. He's back.
Okay. No, I was just explaining that there was very interesting science that was presented here. And the challenge that I was explaining is that we now need to take out the science to the field or to practice, so that we convert the science into products, into goods and services that can solve societal problems. So we really appreciate this cutting edge science that we had today, and we hope that it's going to result in further work, the application work in the various sectors that they are dealing with, these presentations. And I wish all the best of luck as they continue with their work. Thank you very much.
Thank you, all the speakers, and also the moderators and the organizer. And it's a great session. Thank you. Have a good day.
Thank you.
Thank you, speakers. I would like to thank the chairs and also the speakers for their time, interesting contributions. Thank you to our attendees. I hope you found the webinar interesting. As a reminder, please note that the recording for the webinar will be available on the website as soon as possible. I wish everyone a nice day or evening, depending on where you are. Bye-bye.
Bye-bye.
Bye.